Okay, so in the electron transport chain, we saw how both NADH and FADH2 result in translocation or pumping of protons from the matrix to the inner membrane space. NADH moves its 2 electrons through the electron transport chain and results in the translocation of about 10 protons. These 10 protons lead to one full rotation of the ATP synthase, resulting in the production of about 3 ATP, one for each alpha beta dimer. FADH2, due to how its electrons move through the electron transport chain, only results in the production of about 6 protons, okay? This gives us about 2/3 of a rotation of the ATP synthase and about 2 ATP synthesized per FADH2. Now, what we didn't see together in this class is that the proton gradient is used for more than just the rotation of the ATP synthase. There are a wide variety of other transporters that use the proton gradient to get other things into and out of the mitochondria. And so what this means is that in reality, only about 2.5 ATP are made per NADH and only about 1.5 ATP are made per FADH2. The last thing to note here is kind of a big picture idea that we'll talk about more together. So proton uncouplers are an interesting type of molecule to think about with electron transport and ATP synthase, because what they do is dissipate the proton gradient, or you could say eliminate the proton gradient so that the concentration of protons on either side of the matrix is equal. The result of using a proton uncoupler is that ATP is no longer synthesized. So if you eliminate the proton gradient, no ATP is synthesized. This makes a lot of sense, right? If the protons can't move through the C ring, there's no rotation, therefore no ATP is synthesized. And so we're going to think a lot about molecules such as proton uncouplers. And we'll think about how changes like using a proton uncoupler will affect all of the metabolism we've learned so far, glycolysis, citric acid cycle, etc. So what have we seen, folks? This is the end of oxidative metabolism. We have seen glucose going through glycolysis. We've seen how pyruvate from glycolysis is turned into acetyl-CoA, which goes through the TCA. The TCA gives us NADH and FADH2, then move their electrons through the electron transport chain, eventually using the proton gradient to synthesize ATP. Okay, folks, on the last day of oxidative metabolism, we saw Coulomb's law is still important, right? I've mentioned it throughout this class, so hopefully you saw how it's kind of an overarching theme that we can use to understand a wide variety of biochemical subjects. We saw, finally, why NADH and FADH2 are considered high energy. Their energy is used to make an electrochemical gradient that directly results in ATP synthesis. Now we saw a kind of cool example of this because we saw how the proton gradient physically moves a protein. So we call ATP synthase a molecular motor because it is physically moving to synthesize this ATP, which is super cool. And that wraps up our discussion of oxidative phosphorylation. Thanks, folks.